All right, well, it looks like we have a critical mass here, and we'll keep letting folks in as they join. We have about 130 people who signed up for today's webinar, or forum meeting, and hopefully we'll be able to they'll be able to capture everything. We will be recording today's meeting, as you may have noticed, so we'll share it online afterwards. And we have a relatively full agenda, so we'll go ahead and jump into things. Today, we're going to be talking about this multi-stakeholder forum for advancing equity and safety in pulse oximetry. I'm Michael Lipnick. I'm an anesthesiologist. I'm an intensivist at the University of California, San Francisco. Also help lead our WHO Collaborating Center and our Center for Health Equity and Surgery and Anesthesia. And I'm an investigator in the UCSF Hypoxia Lab, which is helping to lead the open oximetry project, which I'll tell you a little bit more about as we progress. I have no disclosures, and here's today's agenda for what we plan to go through over the next 2 hours. Hopefully we'll be able to wrap up a little bit sooner than that. I know everybody has busy schedules. There will be 3 sections of the discussion today, or presentations rather. Each will be followed by a brief discussion, but we're going to be trying to do a lot of interaction in the chat and also interaction in the shared Google document, which Fifi has put in the chat window. If you're unable to access that, I know some folks can't access Google Docs, please put your comments in the chat, and we'll also happily email you the copy of this document as a Word document. We will also be posting the comments online at some point, so you'll be able to see them in multiple ways so that everybody has access. Just a few ground rules before we get started. This is set up as a regular Zoom meeting, so you can take yourself off mute, you can talk, you can use video or not, up to you. We encourage everybody to, when they're talking, at least have their cameras on. The session is being recorded, again, just to flag that. But please do remain on mute aside from the discussions when you're talking. In future meetings, we will attempt to provide live translations for the meetings into at least French and Spanish to try and diversify our audience. We're very open to suggestions on other languages. If there are colleagues that want to participate from other countries, please let us know and we can try and arrange that ahead of time. But unfortunately for today, we're all in English. But thanks for bearing with us on that. So I'm going to start off with some brief introduction objectives about this forum and some background before handing over to our first speaker. Everybody's probably well familiar with a lot of the issues that are outlined here. Clearly has been a skyrocketing number of pulse oximeters entering the market, many with variable performance and quality over the last 10 and especially the last 5 years. During the pandemic, thanks to work from several groups, especially the Michigan group and Dr. Schoeding and others, there was renewed attention to this issue of the impact of skin pigment on performance. This recognition of health and healthcare disparities that may be resulting was a relatively new finding, although the challenge has been lurking for quite some time. Research has been really skyrocketing in this area as well. The number of publications in the last couple of years on this topic is more than the number of publications in the last 30 years on the topic. Also happening during COVID, large-scale procurements. This may have been by ministries of health, hospital systems, donors, trying to figure out which devices work and which devices don't, which devices might be appropriate for certain settings, et cetera. In addition to the procurements, and thanks to COVID, there's a lot of attention to hypoxemia. Pulse oximeters now a household item. Almost everybody knows what a pulse oximeter now, or certainly many more than did prior to the pandemic. And this has resulted in a number of initiatives to try and address some of the challenges that exist, but often these efforts are in parallel. And just flagging this last point, while there's been a lot of attention lately to performance, quality of devices, which is an important topic, this should not be overshadowed by the bigger issue in many ways, which is global access. Access to pulse oximetry is still a problem for the majority of the world, and keeping that in mind as we're addressing solutions to try and improve the quality of devices on the market is essential. For those of you who have not heard anything about the Open Oximetry project, I'll try and keep this brief, but encourage you to read more about the project online. The Open Oximetry project came about because of many of the issues that I just shared in the past slide. This is a collaborative effort of the UCSF Hypoxia Lab, our Center for Health Equity and Surgery and Anesthesia, and many other collaborators. It's built off of work that the team's been doing for more than 40 years. but really was influenced and accelerated by some of the factors on the slide prior and the pandemic. The first component of the project is a prospective clinical trial in critically ill adults, trying to understand the magnitude of the effect of skin pigment and other factors on oximeter performance. This study is being done with support of the FDA, and you'll be hearing more about this study specifically later in the talks. We're also doing healthy human lab-based studies to test performance of commonly available devices. Our lab does that for sponsors, for companies, for developers who are making devices to come to market. But as part of the Open Oximetry project, we're going to be independently testing a variety of devices that are commonly found, especially those that are more accessible in low and middle income countries. We're also working to develop in vitro methodologies that can try and help test device performance, stress test these devices in less resource intense ways. We're also working to create new techniques and validation protocols. A lot of the focus for that currently is how to quantify and account for skin color and device validation. We know many groups are working on that in particular and are eager to engage further. The last two pieces here, which have a lot of relevance to this group, are creating an open access online oximeter performance repository, data sharing platforms, as well as a repository for just information, frequently asked questions, publications, and knowledge sharing around pulse oximetry. And as relates to this forum, most directly is collaboration. We know there are lots of groups that are doing this. We have been in touch with many of them. Many have reached out to the lab, and we are seeing lots of opportunity to try and promote communication and bring together stakeholders from diverse disciplines, geographies, industries, viewpoints, to try and cut down on duplication of efforts and share knowledge, really accelerate progress. The reality is there's just a lot happening, especially in the last 2 years in this space, and we think that things could move a lot quicker, save everybody time, and have the impact that we're all aiming for if we can try and organize the science a little bit more. So with that in mind, jumping into some of the objectives for this particular forum. Some are quite lofty and may seem perhaps unattainable in some ways, but some are going to more clearly seem achievable and with immediate impact, I think, for a wide array of stakeholders that are on this call. First, we're just trying to map who is out there and doing what. We've already started working on this. Thank you to all those who signed up for today and who signed up and provided information in the Google Sheet. Again, we'll share that. That's accessible in the Google Doc as well. So you can click on that link and see who's on the call today, see bios and what people are working on, their backgrounds. So far, we have about 100 people who've signed up. We have an additional list that we'll be merging in of another 40 initiatives that are working on projects at the intersection of pulse oximetry and health equity. So please keep an eye on that. Also, please share in the chat or in the Google Doc additional teams, individuals who you know of that might be working in this space so that we can reach out to them and try and make some connections. There are a lot of groups that are convening stakeholders. We are trying to find ways to link with these committees and initiatives directly and really trying to avoid becoming another parallel meeting. We know everybody has, many people are participating in these other groups as well. And so we're not trying to create another parallel initiative. And we are having positive conversations with several groups to try and figure out how we could streamline, cut down on the number of meetings, perhaps jointly hosting meetings on certain topics and promoting some crosstalk between these. A lot of the initiatives are focused on and are led by high-income countries. And so we also are hoping that this particular forum can help diversify the dialogue and link expertise and perspectives from low and middle-income countries as well, so that all the work that's being done can have the kind of positive impact on the broadest population possible. Another objective for the forum is really refining the problem list. There are it seems every week there's additional challenges that are identified and research questions that are coming up. Clarifying what the problems are and what the priorities are is going to be a big part of what we hope this forum could do, possibly even using some sort of an additional Delphi process and follow-up where we would actually, as a group, come up with consensus on what the priority list is, not only to make others aware, but to try and coordinate among different groups that are working on to prevent duplication. some of the buckets of topics that have come up, there's the issue of performance, certainly, and performance problems leading to disparities and safety issues. There's also there are also challenges related to procurement, leading to inappropriate purchasing or allocation of devices, and then also implementation of pulse oximetry, education, communication, provider awareness. These are all issues that have been highlighted by either in the survey that we conducted or people reaching out to us or our own observations, but we think there's a lot of room to expand on these. We will be inviting input on priorities. Later on, we'll come back to this and have some discussion about what you think are priority areas for inquiry, for action as relates to improving equity and safety in pulse oximetry. Please include, add to the chat or add to the Google Doc. There is a section inviting input on priorities where we're listing these priorities out. Again, we likely will use this as the foundation for a follow-up survey where we'll ask people to help prioritize and rank what they see from their experience and perspective as being items that should be prioritized globally on the agenda. The last objective that I'm going to mention today is opportunities for harmonization. I already talked about streamlining and linking overlapping parallel initiatives, but there are a lot of other areas where we think harmonization could really accelerate progress and benefit many of the stakeholders that are on this call. One has to do with protocols. This includes study protocols for clinical studies, protocols for lab-based validation standard protocols, quantification of skin color, perfusion, et cetera. We've begun sharing all of the study protocols that we've been working on online. So on the Open Oximetry website, on the study protocols page on that, you will find Google documents that have all of our study protocols there. They're all open for feedback. So there are a couple of ways to give that feedback. One is you can add comments directly in those documents, and we'll consolidate and provide a venue for discussion. We'd love to hear what other labs and teams are doing and to try and refine these to not only make sure that we're all collecting the same data so that it can be comparable and maximize its utility, but also make sure that we're collecting the right variables. So please check those out, and we will come back to that after one of our talks later, more specifically around one of the higher priority areas at the moment, which is harmonizing the collection of prospective real-world clinical trials, of which there are several ongoing, and we'd like to try and make sure that we're all collecting comparable data so we can really answer these questions as quickly as we can. I've kind of already touched on this issue of establishing best practices for data sharing and data collection, as well as this idea that promoting clear and consistent communication among the different groups that are working in this space will be really important for some of the primary stakeholders, which are the end users of pulse oximeters, whether that's a clinician or a patient. That is another area where we hope this forum can help harmonize some of the ongoing initiatives. And then finally, harmonizing different standards for pulse oximetry. And I use the word standards relatively broadly as a non-regulatory based person, so please forgive me on that. But there are a lot of stakeholders that are issuing guidance or specifications on pulse oximetry, and there are differences between those. There may be potential to try and identify common requirements and integrate those more readily into some of the more widely used and shared standards to try and cut down on the number of parallel documents that are out there that make it somewhat tough to navigate for some stakeholders. The overall idea here is that we're really trying to coordinate the science, make sure that we engage a diverse enough, a broad enough stakeholder group, and that really will provide the foundation for policy, for advocacy, for impact, and what we're all trying to do. We are a bit worried that without that, it's going to be challenging to get where we want, and that can't happen only at the 30,000 foot level. It needs to engage really with grassroots, frontline providers, patient communities. It can't all happen in high-income countries. It can't all happen in academia or industry. We see a lot of potential to really try and bring these groups together. So with that, there are some very specific benefits that we hope that participants will be able to appreciate and are intentionally trying to design this so this is not perceived as an impediment or a competing body or a threat to any groups. And that really has benefit for everyone. So, of course, there'll be interaction with experts and that you may not otherwise come across, and the potential for new collaborations. Again, this idea of creating concerted voice, consistent messaging. We do hope through this forum that there will be opportunity that folks will be willing to share preprint data, discuss findings of studies as they're happening to try and accelerate everybody's work. That obviously be a voluntary component of this, but something that certainly our team and the Open Oximetry Project will be most certainly engaging in doing. We also, again, will hopefully be able to co-create some of these protocols to harmonize the data collection and improve the interoperability across sites. And then lastly, accessing testing resources. We're looking at ways that we can leverage our lab infrastructure as a global good, and hope to be able to report back on that in future meetings. So to wrap up this brief introduction, here's some of the slightly less lofty objectives for today. Again, today is really an introduction to this forum. We don't expect to leave today and have figured out the answers to most of the questions that have been posed before. But a primary objective, in addition to just introducing the forum, is that we want to just start this dialogue and figure out who's doing what and try and start putting on the map what the priorities should be. You will see after subsequent speakers opportunity for some of that discussion to take place. And again, please continue using the discussion chat and adding in the Google document. At the end of this meeting, I will share the next steps, but briefly for those who aren't able to make the entire meeting, we will be following this up with a workshop in February of 2023, where we'll have subgroups based on the priorities that are highlighted today to try and dig deeper into some of those topics, make some progress. There also will be surveys that come in, or a survey that comes in the interim to help kind of refine the goals. And we really want to invite input on how to make this forum maximally useful to everybody who's participating, because we know time is limited and we appreciate your time. So back to today's agenda, I'll be handing over to our first speaker, Dr. Melvina Edelman, and I will go ahead and stop sharing my screen during the introduction, Dr. Edelman, so that you're able to share your screen. And I will make you a co-host so that you're able to do that. While Dr. Edelman's pulling up her slides, for those of you who don't know her, Dr. Edelman is currently the director of the Office of Ophthalmic Anesthesia, Respiratory ENT, and Dental Devices at the U.S. Food and Drug Administration. For over 20 years, she's led multiple divisions and initiatives at the FDA and has played a key role in assuring safety and effectiveness of medical devices. She's guided development of more than 50 international and national standards, has overseen the development of numerous regulations and guidance, and convened over 30 public meetings of the FDA Medical Devices Committee. She has originated numerous symposia, workshops, organized multiple multi-stakeholder public and private partnerships, and spearheaded many clinical and laboratory studies to improve safety of medical devices. She received her MD from Harvard Medical School, as well as a doctorate in health sciences and technology at MIT, has multiple patents, has published over 100 peer-reviewed publications, and we're very honored to have her speak today. Thank you, Dr. Edelman, for taking the time. We can see your slides, although not yet in presenter mode. Thanks, Dr. Edelman.
Can you see it in the presenter mode now?
All looks good. We can hear you well.
Okay, fantastic. Well, thank you again for the introduction. I'm very excited to be part of this groundbreaking meeting. I'm just going to take a few minutes not to take away from the key points of the purpose for today, but I just wanted to give a very broad introduction of FDA's role in pulse oximetry. And my slides don't want to advance. Okay, just give me a minute. Not quite sure why. Okay, let's try this. Okay. So in the United States, all of the pulse oximeters are regulated as either over the counter, or as we refer them to OTC pulse oximeters. And these are most commonly intended for general wellness or sporting aviation users and are not intended for medical use. They are often sold directly to consumers in stores and online. Now, for the majority of the meeting, I believe this committee will be discussing prescription use pulse oximeters. These are used to monitor oxygen saturation level of patient, most often in hospital settings and doctors' offices, although they sometimes may be prescribed for home use. They're reviewed by the FDA through what we call a 510(k) pathway, and they do undergo clinical testing to confirm their accuracy. As Mike already referred to it, there are many standards, but the one that we do recognize as an FDA for pulse oximetry is ISO 80601-2-61, and I believe there will be a later discussion further on that. Additionally, we have issued a guidance in 2013 that delineates some of our thoughts and recommendations for assessment of pre-market assessment for pulse oximetry. Furthermore, the class 2 devices are regulated under VIA Pro codes. And for those of you who are interested, the regulations and the Pro codes are on this slide. I'm sure everybody on this Zoom call is aware that there are many factors that impact the accuracy of pulse oximeters, which include skin pigmentation, dyshemoglobinemias, anemia, perfusion, dyes, nail polish, ambient light, etc. Most importantly, the scientific community has been aware for some time that certain patient factors may impact the accuracy of pulse oximeters, and one of the factors that we've been concentrating on includes skin pigmentation. Between 2013 and 2020, there was some limited literature on the subject, but in 2020, as was already alluded, the COVID-19 pandemic brought increased awareness of the importance of pulse oximetry, both in hospital and home settings. In December of 2020, Shorting published an article, which all of you I'm sure have read, which reported that Black ICU patients had approximately 3 times the rate of occult hypoxemia compared to the white patients. This article received media attention and spurred additional interest in related research studies. We at FDA wanted to provide the public with information on the interpretation and limitations of pulse oximetry. And so in February of 21, FDA released a safety communication with recommendations for providers and patients. At that time, we committed to continuous evaluation of the safety and effectiveness of pulse oximeters, including a review of relevant literature. This literature was indeed reviewed and has demonstrated mounting real-world evidence from studies capturing tens of thousands of patients that pulse oximetry accuracy does indeed vary by self-reported race and/or skin pigmentation. However, there is still a need for prospective studies that utilize standardized measurement of skin pigmentation that capture simultaneous measurement of SaO2 and SpO2 pair data, and that systematically collect data on important confounders in order to have more robust evidence about the impact of skin pigmentation on real-world oximetry. On November 1, 2020, just about a month ago, we at FDA convened a virtual public meeting of the CDRH Anesthesiology and Respiratory Therapy Devices panel of the Medical Device Advisory Committee to share information and perspectives from interested parties about ongoing concerns that pulse oximeters may be less accurate in individuals with darker skin pigmentation. We have delineated our questions to the panel in the following 5 panel questions that I'm going to just skim through quite briefly. In the first question, we asked the panel members to discuss the clinical evidence from the scientific literature about the accuracy of pulse oximetry among patients with darker skin pigmentation. The second question was was focused on tools to assess skin pigmentation. And we asked specifically for recommendations for studies evaluating pulse oximeters on standardization of skin pigmentation assessment, categorization, and reporting of this data. In question 3, we talked about ARMS, the root mean square of pooled data pairs that we usually use. And we asked the panel to comment on the accuracy of pulse oximeters. Question 4 dealt with labeling and the panel's recommendations on how we should label and communicate what the pulse oximeters do for both OTC and prescription use. So we had a very, very productive, very full day meeting on November 1, and are still trying to delineate the exact steps that FDA will take to address all of the panel recommendations. However, we know that we can achieve better outcomes in protecting public health when we integrate the different perspectives, experiences, resources, and expertise from each participant in medical device ecosystem. One way to bring these voices together is through the collaborative community. Collaborative community is a global grassroots effort convened by interesting stakeholders that works to solve shared challenges and achieve common goals related to medical devices. Now, collaborative communities are not convened by the FDA and not led by the FDA. The membership and governance are not directed by FDA. However, we do participate if asked to, we do participate in collaborative communities if we believe it addresses the health, public health issues that are important to our mission. Now, what makes collaborative communities different than task force that everybody is very familiar with? Again, the collaborative communities are convened by external partner, not by government entity. And they focus on a broader outcomes and impacts, and they have multiple deliverables for each work stream versus the typical task force, which provides a specific recommendation to address a single issue of concern. Collaborative communities are envisioned as an entity with sustained duration and a potential indefinite lifespan. Also very important that all stakeholders are brought into the effort and have an equal voice in the decisions. To help those who are interested in this concept, we put together a collaborative community toolkit that is available on our website on this slide. We also have on our website delineated best practices for establishing and maintaining collaborative communities. And as you can see, these do include casting the membership net broadly and strategically. With that, the sectors that we like to make sure that the collaborative community identifies for potential members is very, very broad. And does not necessarily list is not inclusive, is not exclusive of this. In other words, we would like to see as many different partners identified and incorporated in the collaborative communities, especially the ones that we choose to join. Now, what are the potential outcomes of a collaborative community? These could be, and often are, peer-reviewed publications, research agenda and projects, white papers, information and data gathering that will help make determination for healthcare coverage and reimbursement decisions. Often it's best practices or templates or frameworks. We have started this a couple of years ago, and here's the link on our website where you can access all of the collaborative communities that we participate on at this time. Again, as of today, FDA is member of 12 collaborative communities. And these efforts help us address the challenges that have been ineffective to significantly expedite innovation by one small parties. And I just want to highlight that in the area of pulse oximetry, as Mike has very eloquently pointed out before, there are many, many entities that are working often in parallel on the same tasks. And I truly hope that today's meeting will motivate all stakeholders to continue to work collaboratively and to integrate different perspectives, experiences, resources, and expertise so as to allow everyone to prevent duplication of effort and optimize novel approaches. And with that, I want to thank you for your attention, and I'm going to stop sharing my screen.
Thanks very much, Dr. Edelman. I'm going to go ahead and pull up the agenda again. And just to follow up on Dr. Edelman's comments, we are in the process of planning to invite the FDA to participate in this and going through the process that Dr. Edelman referred to on the website, the necessary steps so that we can engage as a collaborating community. So with that, I'd like to open up just for about 5 minutes here for some brief discussion. There have been a couple of comments in the chat, but on this issue of collaborative communities, on the issue of this forum and the background that I provided and the objectives, we would welcome and invite feedback either in the chat, in the Google Doc, or if you raise your hand, we'll go ahead and you can take yourself off mute. But please comment on these two questions. We would really welcome the input. And there's no way that nobody doesn't think there's going to be challenges with this. So,
Mike, I'll try. Do we want us to raise our hand, or do we want us to just speak?
Go for it, Sandy, please.
All right. Well, I'll chime in, and everybody else can raise their hand. Sorry. So, Sandy Weininger, FDA. I think one of the opportunities here is, as you've listed, there's lots of different, I'll call them documents or clinical practice guidelines or engineering standards that are out there, and they kind of all operate independently as their own little silos. And to the extent that we can bring them all to the same place and understand the relationship to each other and what the big picture quilt needs to look like and what role those particular documents play, I think we can all raise the public health perspective here.
Thanks, Sandy. I see Valencia, you have a hand up.
Thank you. Thank you. My name is Valencia Kumsen. I'm an associate professor at Tufts University in electrical and computer engineering. Thank you for the forum. And I just have one question about the open oximetry project. I did notice that there is now an open database of devices that have been tested by your lab. And I was wondering if you have a vision of seeing that as a place where consumers or healthcare procurement offices could go to find data on error data on devices. And also second to that, have those devices been tested on ranges of skin, a range of skin pigmentations? I didn't notice that on the site.
Thanks for the comment. I'll try and address that briefly, and happy to follow up and talk in more depth. Currently, the site is made as a demonstration site. The data are real. We've used the kind of traditional 510K pathway that we test for all devices. So in the sense that there is a requirement for some diversity of skin pigment, we have followed that. So the data that are on there are representative of that. However, we are rebuilding that site, and we'll be launching a new version of the site. There'll be two ways that the portal will be two users. One will be consumer oriented. It'll look somewhat similar to what it is right now, kind of easy to navigate, get high level information. And we hope to expand the protocols that we use to test the devices. So more rigorous testing of skin color, more rigorous testing of perfusion. And so that is kind of coming down the pipeline. But the other portal and the other use case will be, we intend to share all of our raw data. So we're collecting raw data in the lab, data that's beyond what's used for regulatory purposes. And we know that there are researchers who have other questions that could be asked that perhaps we're not thinking of, and we want to make that available so that others can use that data. So that we will also be building. That will be probably second quarter 2023 for launch, but that's where we're heading with that. And then we will be releasing another batch of about 15 devices over the next few weeks, and are hoping to test about 100 devices over the next year and a half. And we have a list that is shared on the website, and we welcome feedback and input. To date, those devices have come from input from ministries of health, aid agencies, and consumers. So appreciate input on that.
Thank you. Thank you.
Thank you. Julian.
Thank you. So I have 3 suggestions for using the forum, or 3 kind of broad issues that I think need to be addressed somehow. One of them is a proposal of research questions. So people who have the resources to perform the research themselves typically won't share research questions because they plan to investigate. But there are plenty of others who have research questions that they think might have utility, and they have no interest or ability to pursue it themselves, or perhaps they want to collaborate with others. So I think a forum to contribute research questions is one area. Another is a way to pose technical questions or clinical questions that, I think many of us are fielding questions, and we're having a lot of sidebar conversations trying to kind of get to the truth, if it exists already, if it doesn't require further work. So I think what could essentially become a publishable, or even if it's just web content, FAQ on pulse oximetry, in which we are asking each other experts questions in sometimes esoteric areas, and get those answers and collate those. So that's the second topic. And the third is, I think that there are various opinions out there about what might be the factors that are causing issues. There are hypotheses, there are opinions. I think some of those people are happy to just put out there and have either considered or debunked. So not a well-formed research question, but an opinion. And I think some people are happy to do that with attribution, and others really would like to do that anonymously. There are people, for example, that work in companies that don't want to speak on behalf of the company, but have deep experience and expertise, and have strong opinions about where the most useful, efforts are, where to really apply our energy and our efforts as a community. So anyway, those are the 3 buckets that I think a forum like this and a community like this could really help capture information, share it, and advance the cause. That's it.
Julian, thanks for the comments. Just a quick response. I think those are all great ideas and ones we hope to follow up on, especially the point of people are going to want to share in different ways, and having a forum a mechanism where people can share anonymously. We're definitely on the same page with that. Thank you again for that. Sarah.
Hi, Mike, and thanks very much for convening the forum. So I guess the one thing I wanted to say, as you pointed out in this, there's a lot of interest. People are waiting for direction as to how to move forward. They're trying to find direction in their own way. And the racial disparity issue is very prominent. And we have an immediate need for clarification and guidance on the best way to measure skin melanin content. Currently, I understand, and I'm not sure if this is true, but what I understand is that the FDA is now requiring Fitzpatrick scale to be used when they have data presented. The Fitzpatrick scale, as probably everybody on this call knows, has been discredited over the last 20 years very significantly, and people do not want to use the Fitzpatrick because it is an instrument that has been used in ways that are opposite to actual health parity. So the Fitzpatrick needs to go now. So what we're talking about is right now we need to have some decent method guidance about what to do with skin melanin content if we're going to do these ICU investigations or other types of stuff. So that's my 2 cents. Thanks.
Sarah, thanks for that. We'll go to Ellis next, and then Eric, and then we'll conclude this. I think Sarah, the discussion on that, I think everybody on this call is going to agree that there's a lot to be done there, but that will be the topic of a very dedicated conversation that will follow, if not a complete subgroup. I know there's a lot of work happening in that space. Ellis, maybe if you could quick intro and then comments, the floor is yours.
Absolutely, yeah. My name is Ellis Monk. I'm an associate professor of sociology at Harvard University. I'm also a visiting faculty researcher at Google. Most relevant to today's discussion, I created the Monk Skin Tone Scale, which has been adopted by Google and some other companies as a measure of skin tone that is an alternative to the Fitzpatrick scale. And the idea here is that, as Sarah pointed out, the Fitzpatrick scale has a pretty troubling legacy, and lots of people have pointed out that it's inadequate. In a paper that's under peer review with myself and some researchers at Google, we have already found some results that suggest that the Monk Skin Tone Scale is more inclusive than the Fitzpatrick. And like I said, it's already been adopted by Google and a lot of the different products that they're using in machine learning and artificial intelligence. So this is an issue that is being worked on in some capacity. I'll put a link in the chat to that project with Google. And my hope is that one aspect of this forum is having a really dedicated conversation about the standards of measurement of skin tone. Thank you.
Thanks, Ellis. And I anticipate that there'll be a dedicated subgroup on this topic that we could convene. Again, a lot of people doing work in this space, and we are collaborating with Ellis and his team to use the Monk scale in addition to several other techniques in all of our studies, and it's evolving process. So you can see the online, there is our current skin color protocol, which uses multiple scales, multiple techniques. We welcome input. We're very eager to collect data that others are interested in and share that data. So please comment there. Eric, over to you. Thanks, Alice.
Thanks, Mike. Hi, everyone. My name is Eric McCollum. I'm an associate professor of pediatric pulmonology at Johns Hopkins, based in South Africa. The issue, I think, one issue that I think would be additionally important for this forum to address is with pulse oximeter testing and development as it pertains to neonates and infants and children. Having worked in LMICs with colleagues over the past 15 plus years, the real common scenario that has been exacerbated by COVID are that there's an influx of devices into LMIC contexts, but they don't really work for kids. And when you look at how devices are developed, they tend to be developed for adults, tested on adults, and then when we implement them in the field, not surprisingly, they don't really work for children, infants, and neonates. And so I think that's an issue that would be, in my mind, important for this forum to address, hopefully. Thanks.
Great point, Eric. Thanks very much. Josh, over to you.
Hi, Josh Pfeffer from FDA. I wanted to just mention that we're certainly interested in different approaches for assessing skin pigmentation level, looking a lot at some of these optical devices that some call melanometers based on colorimetry and spectroscopy. And I actually also wanted to ask Dr. Monk if there's a specific journal article. I was trying to learn more about the scale that you proposed. If there's an article or two that you can put a link to that we can learn more about this approach.
Thanks, Josh. And Ellis, if you could put the article in the chat, that'd be great. And just to flag, we are, as part of the project, trying to create a repository that's tracking different publications relevant to this. We've started with oximeter performance, but the issue of skin color quantification methods, we'll be trying to provide some sort of a repository to keep track of that. Sam, and then we'll take the last comment from Victoria, and then we'll have to move on in the agenda. Apologies. Sam, over to you.
Yeah, thanks, Michael. Hi, everyone. Really quickly, I'm CMO for patient monitoring and Medtronic, and very proud to be part of this work stream. I'm dealing with a lot of customers right now that have real-time, sensible questions about what do we do right now? And I'm sure all of you are dealing with those as well. I think one of the things that we should focus on, much like the article this month in Respiratory Care from Dr. Moore at UCSF, is kind of keeping some flow of information to our partners in the field that are taking care of patients with our devices every day. It's sort of, it was a beautiful editorial and very, very helpful. But I think this group really should think about consistent messaging coming out of this forum, and perhaps even a place online within the structure of this group for people to come and get up-to-date information and advice. We've got a lot of folks looking for protocols, lines in the sand, things like that, and we want to be able to assist, and I think this is a great group to do that.
Sam, thanks very much. Victoria, over to you, and then we'll be moving on in the agenda.
Yes, one thing I'd like to see remain a focus is how this group and this issue can really solve the problem of aid colonialism, where product is dumped and the product may not work. And overarchingly, there is a need for an open source, simple, easily locally manufactured device within this entire field. And I would like to see that, this very encouraging open data, open documentation forum, remains so. And even if a commercial product is brought to market, that we still have an open source baseline, because not every community is well served by imported aid. There are communities all over this world that cannot receive imports. So I would like us to hopefully keep open source as a foundation of this forum, and keep breaking colonialism as a goal.
Thanks very much, Victoria. Thanks for those comments. So we're going to go ahead and move on just in the interest of time. And our next two speakers in the next section will follow with a bit of discussion as well. Our first speaker, Adriana Velasquez, is joining us from the World Health Organization, where she's the team lead for medical devices and in vitro diagnostics. Ms. Velasquez has done an incredible amount of work during the COVID pandemic, and certainly before, as it relates to improving access to devices, educational material, and reviewing emerging technologies relevant to oxygen therapy and diagnostics. Dr. Velasquez, I'll hand it over to you. Thanks so much for taking the time to join us today.
Thank you very much, Dr. Lipnick. So good afternoon, good morning, good evening, everyone. Really, when Mike called me, I said, absolutely, WHO wants to support this work. So this is really a minute to say there is a statement of support for this work. I think COVID showed to all of us how we can all work together and be much more effective on really delivering what is needed in the ground. I think this is an excellent and absolutely incredible opportunity to make really a change for everyone that needs it. So I lead the medical devices team in WHO, and our goal is really to increase the quality, the safety, accessibility, affordability of medical devices. We target also innovative technologies. We look into standards, technical specifications, but really the main thing is to ensure that there are good diagnostics and good tools for those that need them the most. And we recognize the work that is being done by Dr. Lipnick because he is leading the WHO Collaborating Center, and we will support this initiative and basically to streamline and absolutely to not create duplications, because WHO was going to start this work. Then we see the work that is going on, and the main idea is how can we all work together. We are aware also of some limitations, for example, for the ISO committees, that maybe low and middle income countries have no access, but that changed also over COVID when they put their standards out to share by everyone. So I think we are in a time where many things are changing. We now have a STAG, a strategic and technical advisory group for medical devices in WHO. And I think one of these projects like this one that we are here on will be highly prioritized. For us, it is very important in WHO to harmonize the protocols, the validation, the laboratory data, share the nomenclature, share specifications, and have standards that could be used by everyone. So we will share and we will invite everybody from all stakeholders' work. and to agree that really pulse oximeter is an essential basic tool, diagnostic tool that can serve not only pneumonia, but many other diseases. It helps hypoxia, but and that affects many other diseases. It is used from community health services to all the way to tertiary health institutions. So I absolutely agree on participating in this forum to share with all our WHO collaborating centers, to also all our NGOs in official relations that are working with us, that include biomedical engineers and medical specialists, and welcome this initiative. We'll support as needed. We'll be happy to work with this forum, also with the ISO and whoever, and regulatory agencies, procurement agencies. And remember, every time I have my calls, it is remember technology is not the most important thing. What it really, really matters is the patient that is behind the technology. We really need to help those patients, and we can do it all together. So thank you, Mike, for the opportunity, and looking forward to working with all of you. Thank you.
Adriana, thanks very much for taking the time to join and for sharing those thoughts. We'll have one more speaker, and then we'll have an opportunity for comments in just a moment.
Thank you.
Our next speaker is Sarah Liadi Mobaran from UNICEF, the United Nations Children's Fund, part of the Supply Division Product Innovation Center. She's currently the Newborn Health Innovation Specialist at UNICEF, and her center seeks to drive demand and increase accessibility to appropriate and innovative technologies in the newborn space. Sarah has worked as a biomedical engineer and an educator with the Newborn Essential Solutions and Technologies NEST at Rice and Rice 360 Institute for Global Health Sciences groups, with a focus on newborn health technologies. She's going to be joining us today and talking a bit about procurements and how to integrate the issues of accuracy and varied melanin, inaccuracy due to varied melanin and perfusion into procurement guidelines. Sarah, over to you.
Thanks so much, Mike. Sorry, guys, I'm going to keep my video off just for bandwidth issues, but very happy to be here with the forum. Yeah, and we can go to the next slide. I'll be discussing the product innovation work that we're doing, as well as some collaborative work with my colleague, Hawa, who is with the Health Technology Center. So UNICEF has a strong relationship with oximetry, and our priority really at Supply Division is to increase accessibility to the products that we know are usable, and that will create a sort of sustainable supply chain for making sure that there's access to these products. So we're focusing on elements that are robust, low cost, and very human factors oriented in design, so that we can make sure that the uptake for these products is appropriate. Next slide. So there's been a history of innovation in pulse oximetry at UNICEF, and I'm coming from the Product Innovation Center, where we really embrace these kinds of gaps that we see in newborn care and try and work collaboratively with the Health Technology Center to increase accessibility. Next slide. So we have a couple of different areas where UNICEF has really tried to embrace this. So there's the ARETA project, which has tried to co-develop products that aid in pneumonia management, and this includes a couple of different products, a respiratory timer, an automated respiratory counter, and a joint multimodal device that includes respiratory rate and oximetry. And that's really where we see this coming into this forum. Next slide. And in line with that, we've worked with Neonatal Essential Solutions and Technologies to really try and push manufacturers to consider the type of parameters that we're looking at, both for procurement and also for general supply. So looking at developing a target product profile for a pulse oximeter, because we really feel, and I think the forum will agree with this, that the perfect product is not available right now. So together with NEST 360 and Neonatal Essential Solutions and Technologies, we've published this target product profile that looks at different types of characteristics, and you'll see that it looks a bit like a spec sheet, but we're really trying to push manufacturers to create this optimal product. Next slide. So in terms of continuing innovation, and Leaf actually, this is coming up in our next slide, how we build this TPP into something that can really address the kind of challenges that we see in pulse oximetry. We're working on continuing innovation for pulse oximetry at UNICEF Supply. So Next slide. And our efforts in this regard are really around trying to make sure that our specs address the kinds of things that we're looking at as essential in this forum. So things like how we really assess integrating skin pigmentation into the specifications that we're using to help tender for these products, as well as perfusion, which we know plays a huge part in making sure that these products are accurate. And actually, as my colleague Howard and I are both working on a request for expressions of interest to look into what exactly is available on the market, not just for pulse oximetry, but looking beyond that into other multimodal devices, so that we can really assess what's available on the market and see how we can integrate that better into UNICEF supply and into the catalog that we have available at country office and also through procurement services. Next slide. And I know this has been a very brief presentation, but really it's because we know that this forum is integrated around trying to come up with the appropriate specifications, the appropriate sort of changes into the way that UNICEF Supply Division is thinking about the products that we're procuring. And we want to make sure that we're integrating those into our process. So I think Mike will be hosting some conversations around how we better integrate this into the procurement process that we're working through as Supply Division. And we'll really be looking to try and integrate that into the specifications that we use to procure in the future.
Sarah, thanks so much. At this point, we'll open up for some brief discussion here, a couple of questions that are posted here. Also, to draw your attention, in the Google Doc that's shared, we are keeping notes. We're recording this, so we'll be transcribing all the comments into that agenda as well. Julian, you had asked where the comments are going. We're trying to keep up in real time, but we'll also be, after the fact, transcribing everything, so it will be in there eventually. At the top, the list of resources, we've included a link to UNICEF's pulse oximetry target product profile, TPP. So if you want to take a look at it, it is there. But now we'll open up for comments on this issue of how to address procurement ahead of regulatory changes taking place, ahead of devices meeting those new regulatory standards. How should organizations be thinking about their procurement specifications? Any comments? Open the floor. And Sarah, please feel free to add if there are specific other questions that you think we should try and elicit feedback on at this point.
No, I think that's perfect. Thanks.
I'll perhaps suggest something that I just put into the comments. Sorry, I didn't raise my hand. There we go. It looks like this, we haven't, one of the things we need is more real world evidence. And of course, ideally that evidence is obtained thoughtfully and comprehensively. But it seems like now that there is this ramp up and the discussions about increased usage and interest globally, especially in patients with darkly pigmented skin, want to capitalize on that and start collecting useful data, whatever that looks like. That's what this thoughtful group could help think about.
Thanks, Julian. I see a comment also from Mike Ruffo in the chat. Mike, do you want to expand on that?
Yeah, thanks, Mike. Hi, everybody. Yeah, we just had a meeting with stakeholders from 6 different countries talking about pulse oximetry in primary care. And they all recognize the value, of course, but as they look to procure, they were asking me, which devices should they look for? Are there technical specifications that they can use to conduct a procurement that for appropriate devices? And yeah, and I know the WHO and UNICEF have published technical specifications recently. I do think there needs to be updates though, so we can be sure that we're addressing the skin pigmentation issues. So I think there's or a list of devices that have been tested or pre-qualified, which I know you're working on, Mike. So try not to rush you.
Thanks, Mike. Yeah, I mean, I think your comment's a good one, which is, there are many stakeholders that need to procure devices now, and the guidance is not going to happen immediately. And so, what is an interim solution? I see a comment from Helen. Helen, do you want to chime in?
Yeah, thanks. I think something that we've seen, and maybe this is related to more procurement lists than regulatory approval pathways, but as some of these improvements on products are more through software, algorithm, AI-based sort of improvements rather than hardware. How do we update these lists or expedite the availability of newer models or newer products on these procurement lists, so that the latest products don't have to take another 2 years to get sort of approved and added to accepted lists?
That's a great point. Helen and Mike, do you mind just briefly, quick introduction for just if you could state where you're coming from for folks?
Yeah. So I'm Helen Story. I'm a public health scientist at PATH and specifically work on diagnostics and medical devices. And we are working on a project looking at scaling pulse oximeters, but also advancing the market for next generation pulse oximeters, which have additional clinical measurement features and or maybe moving into the smartphone based or consumer product category.
Yep, and I'm a co-conspirator with Helen. I also work at PATH, working on the Tools for Integrated Management of Childhood Illness project, which is essentially looking to provide evidence to scale pulse oximeters in primary healthcare, low and middle income countries. Thanks, Mike.
Thanks, Mike and Helen. Appreciate it. Any comments about Mike and Helen, what they've mentioned? Valencia, do you want to comment on that?
Yes, I just had one question. I am familiar with the guidelines set forth by the US FDA in terms of pulse oximeter testing for approval, but can anyone comment about different requirements from other regulatory bodies across the world around pulse oximeters that we should, that in comparison to the current FDA guidelines?
I don't know if Sandy or Bob or...
Yeah, I heard silence for 10 seconds. I was waiting to see if somebody else would chime in. So to the best of my knowledge, this is Sandy Weininger, FDA. And Bob and I are the co-chairs of the ISO-IEC co-oximeter committee or pulse oximeter committee, which we're going to talk about in a moment, as well as, we deal with in the FDA topics with oximetry. To the best of my knowledge, FDA is the only regulatory body that has a recommendation or guidance document directly aimed at the design and construction of devices. There's lots of clinical institutions that have clinical practice guidelines that talk about how to use oximeters as well, in contrast to how to build an oximeter, if you will. But in most of the other regulatory schemes in the world rely on evidence of conformance with the standard. And I'll put a caveat in there. Most of the other schemes of the world, there's two aspects of, if you will, public health. I mean, there's the aspect that the device is safe, and I'll use safe with a capital S because that means lots of different things. But then there's aspects of effectiveness or even efficacy, where you can use the device in a safe, in a proper way for clinical use. And so you need to distinguish when you say, are there other means for, if you will, procuring or, assuring quality of these devices, what you really mean by that. Over.
Thank you. Thanks, Valencia, for the question. Thanks, Sandy, for the comment. Duncan, good to see you. Floor is yours.
Thanks. I can say a little bit about the UK and Europe regulatory system. I've just switched jobs. I'm no longer the clinical director for medical devices at MHRA, which is the UK regulator, but I work for a notified body in France now. And what Sandy said is essentially right, that the European and UK regulation essentially says that if a device is safe and conform with the requirements, then it can be on the market. And in this case, that ISO standard is a harmonized standard. Therefore, it grants you sort of if you conform with that standard, if you meet that standard, you can assume that you conform with the European regulation. But I think the bit that's different is that, particularly in the UK, we don't just rely on the regulation to decide what's available in the National Health Service. So the use of health technology assessment and using sort of contract and procurement mechanisms to control the supply of devices in the NHS is much more common in the UK, because we have got a National Health Service where there's a lot more sort of centralised procurement. Having said that, I think that's been historically quite a weak mechanism for this sort of device, because it's sort of everyone looks at it and goes, well, it's a pulse oximeter, of course it works, just buy the cheapest one, and that way we get the most for the least money. And I think we've realised, particularly with COVID, that isn't a good enough way of doing it. So in the UK, there is now an inquiry going on and some work going on in the sort of the NHS side, so the procurement side, as to what standards with a small s should be applied in the NHS choosing which devices to buy, rather than which devices are legally allowed to be sold in the UK.
Duncan, thanks very much for the comments. I think there's a lot of, I could easily see this being another subgroup that kind of forms the discussion around this. Appreciate Sarah and Adriana sharing. And again, the TPP is in the link, the resources link. So please check that out. If you have additional comments, feel free to add them to the Google Doc or email them to us, and we will circulate, and stand by for other communications around what this issue of what to do while we're waiting for regulatory changes and for those to be implemented. In the interest of time, we're going to have to move forward in the agenda. So Sarah, please throw your comment into the chat for now, and we'll try and come back to it. So next in the agenda, we're going to be hearing about standards for pulse ox, better defining some of the problems as we talk about harmonizing approaches, and that will be followed by a talk about this issue of harmonizing the prospective clinical study data as soon as possible, because many studies are ongoing. So with that, I'm going to go ahead and stop sharing my screen while I do introductions, and Sandy and Bob, you can go ahead and share your screen to get your slides up. We'll be hearing from Dr. Sandy Weininger from the Office of Science and Engineering Labs, the FDA, who, as he had mentioned, is also the co-convener of the ISO-IEC Joint Working Group on Oximeter Device Standards. Dr. Weininger is a senior engineer. He's been working at the US FDA Division of Biomedical Physics, the Office of Science and Engineering Laboratories within the FDA Center of Devices and Radiological Health since 1990. His current scientific and regulatory focus is safety and performance assessments of sensors and actuators for physiologic systems, and examining complex systems to understand unsafe interactions. He works with standards development organizations, very long list of acronyms here, with which I won't botch stating all of them, but he's actively involved as well in teaching, delivering courses on safety of medical devices and engineering principles, and very pleased that he's able to join today and share some of his thoughts, along with Bob Kapotek, who is a dual licensed clinician, registered nurse, and a respiratory therapist, and also a co-convener for the IEC ISO Joint Working Group on Oximeter Device Standards. Bob is currently employed by Edwards Life Sciences as the Director of Clinical Affairs for R&D Critical Care Team. Of note, Edwards doesn't manufacture a pulse oximeter. Bob has worked with multiple employers, including 4 pulse oximeter manufacturers in previous employment. And we again welcome Bob and Sandy to share today. Thanks for taking the time. Over to you.
Mike, thanks. I didn't actually mean you to read the whole thing. You should have just picked out the highlights. But
everybody wants to know who's here. I couldn't fit all that in the Google Doc. So,
right. So this is the Bob and Sandy show for just a few moments. We're just going to try to give you a highlight for what this standard is that we are trying to shepherd through, and try to give you some sense of what it can do, and importantly, what it can't do and what it doesn't do. All right.
Sandy, so are you able to share your slides?
Am I not?
We don't see it yet.
Well, that's interesting. My system says I'm sharing.
We just see your smiling face with a flag.
You
over
see
it.
my smiling face?
Well, with, but yeah, it's not.
Photo,
Oh,
just your photo.
not the actual camera. All right, well, let's push the button again.
Okay.
Now we
Now, do you
your
see something?
Okay.
Slides.
Yeah, we see it. There you go. All good. Thank
Well,
you.
that was interesting. All right, well, let's try this again then. Okay, all right, so first of all, who is this ISO IEC group? It's a joint working group between ISO, International Organization of Standards, and IEC, International Electrotechnical Commission. Each of these standards bodies you probably know or have seen them, but they each have slightly different jurisdictions when we come to the development of voluntary consensus standards for medical devices. This happens to be what's called an ISO lead. It is a joint working group, but it's an ISO lead, which just means that the ISO organization's processes and procedures takes precedence over the IEC organization's equally bureaucratic chain. ISO, we have approximately 90 registered members in our committee. In IEC, we have approximately 39 members. Those are representatives, technical experts, regulators, clinicians, all kinds of different people from the different countries. There is overlap between those two, so it's not like we have 129 independent voices heard from. And members, as we said, represent their clinical professions. Members participate by petitioning their national body to be accredited, to be credentialed to work on the standard. So that's a fancy way of saying you can't just necessarily show up at the meeting, although if you do show up at the meeting, Bob and I are happy to receive your input. But if you want to vote on the particular standard, you have to be properly accredited because each country has one vote. And the attempt is to develop consensus across all these bodies. And so there's a lot of work that goes on within a national body. And in the US, ANSI, which is our American National Standards Institute, delegates that responsibility to AMI, the Association for the Advancement of Medical Instrumentation. They hold what's called the secretariat for this particular ISO committee. Turns out they also hold a secretariat for the IEC committee. So it's very convenient that we have the same people that manage both aspects of it. But I think you get an idea for the amount of bureaucracy that's kind of going on here. Happy to answer more questions about that, although that shouldn't be what our focus is today. Pulse ox came out in roughly 1982. There were various different kinds of things out there. The HP ear oximeter, which weighed a lot and cost a lot and heated up your ear and was not actually a pulse oximeter, right? It was just a standard oximeter. But roughly 1982 is when the first things that came out that most clinical organizations could afford. And since they were non-invasive and seemed to report one of the important vital signs, everybody jumped on the bandwagon. Whoa, there's timing. Sorry, started using them. Very shortly after that, the American Society of Anesthesiologists came out with, their initial findings. Blood oxidation during oral anesthetics. You should assess the concentration of oxygen, right? So the clinical associations said, we got to figure out how to use these properly. And very quickly after that, the standards organizations said, okay, now that there's a bunch of these devices out there, what does it mean to be a pulse oximeter, right? And that's kind of what the objective of a standard is to define, and more technically, it's called basic safety and essential performance. And those have very definitive engineering terms, which we can talk about later. And you can read the list of advancements or engineering and clinical oversights that have been occurring. So what do we mean by safety here? In the construct of a standard, there are quality system standards, and in this case, ISO 1345. There are risk management standards, which talk about how you identify the risks and what you should do to mitigate those risks. And importantly, it doesn't say, here is what we mean by acceptable risk, right? That's the domain of the developers of particular standards or particular users of the standard to figure out what they think safety is. And clearly that's one of the main topics that we have to discuss. What do we mean by safety? And then there's In the below right corner, there's IEC 60601 is the general standard for electromedical devices. So basically all devices that have electrons flowing in them shouldn't shock you, shouldn't burn you, shouldn't crush you, shouldn't have sharp edges. You should be able to read the markings, right? When you plug it into the wall, it shouldn't explode. Things like that, which we think should be common safety aspects around the world. We should all be able to agree that a burn is a burn. Bob, I'm going to turn it over to you now for some background on the oximetry stuff.
Okay, so are you going to the next slide? Because that
I am on the next slide, I believe. The primary paths of light loss in tissue.
No, thank you. All right, fine. So the key point to the use of these devices is that they are attempting to see a change in attenuation of light, so a degradation a bit that is in some modulated time series. And that modulation is then assumed to be the pulse rate. So you can imagine that as this vessel is expanding and contracting, that it absorbs a different amount of light. So that's what's shown there at the very top with what is referred to as a PPG or photoplethysmograph of what that change in light attenuation is about. So that's what is displayed then as pulse rate. Now, the light as it enters and then ultimately exit is lost by two primary methods. About 95% of it is lost to just scattering. It goes all over the place, and the more that it's exposed to fat or especially air, the more likely that scattering is going to occur. And then the other component is absorption. And absorption is based upon other materials that will hold that light for a while and reduce its intensity before it comes back out to the surface where it can be measured. And the principle here, if you go to the next slide, Sandy, is that different wavelengths of light, you see it at the very top there, one in red and the other one in infrared. So it's shown there as kind of a light blue and a bright yellow, that as it impacts the vessel, as it changes in diameter or in volume, in other words, that it actually reduces the amount of light that is then detected on the other side. Now, this is a typical finger-based type of sensor, so this would be a transmittance or transillumination style. But you can see that that attenuation is also there of light because of those wavelengths. And the way each wavelength is impacted differently depending upon the color of blood. So this then makes up the second estimate of what this thing is supposed to read out. And currently there are only two things required under the IEC ISO standard, and that is a display of pulse rate and a display of SpO2. And the SpO2 is assumed to be, as noted in white in the lower left-hand corner, to be arterial oxygenation. So that said, next slide, as you're putting in all of this light and you're only getting out less than 5% of it, there are a lot of reasons why these things are challenged. And one of the reasons why myself and many others have authored clinical practice guidelines so that people that are in the trenches of trying to make sense of what this value is displaying are able to understand other things that they ought to be doing to verify that this estimate of color change with hemoglobin is realistic. Now, the first one is you would sample a direct blood specimen. So that is common practice, but just bear in mind that it really emphasizes the point that this is a non-diagnostic device, in spite of the fact that some societies have taken it on to actually put hard numbers on where intervention or care thresholds are then established. So that is outside of the of our conversation here, but it is certainly something that is a bit worrisome for a device that was never intended to be a direct measure of oxygenated hemoglobin. All right, so that said, obviously you don't have everyone with an arterial access, and you don't have an analyzer that will do direct blood analysis at the bedside of many situations. So pulse oximetry has come in handy, and certainly for even home surveillance of various therapeutic needs, including oxygen therapy, as well as understanding the degree of how COVID might have impacted your lungs. This can be of tremendous benefit. All right, so we've talked about clinical use guidelines and how these societies have tried to inform, but we still need to do a better job of education. And that's true of the standard that we're working on as well. And it behooves the manufacturers to make sure that what is in print with their instructions for use is not only for a biomedical engineer or a procurement individual that has a deep understanding of how devices operate, but as well for somebody that is a lay user. So that is certainly something to be added to the next iteration of the standard that is in process. So I'll leave it at that. I think there's, Sandy, that's back over to you, except my emphasis here, which is.
Right, this is just to give you some evidence of Bob's background. This is Bob. So when he says that he's been deeply involved in the development of oximetry, he means it.
Okay, thank you, Sandy. That's okay.
All right, so I just want to spend, because we're running out of time, I just want to spend a few moments giving you a flavor for the kinds of things that are in the standard, and importantly, what are not in the standard, just so when we start to think down the road of tools, either safety tools or procurement tools, whatever kinds of tools you can think of, you know, we have to understand what documents hold that kind of information that we need. Okay, so as Bob said, the pulse oximeters are about the estimation of arterial hemoglobin saturation, right? It's an estimation, and as well as pulse rate. And importantly, if this little last clause in yellow, it says hazards inherent in the intended physiological function, this is trying to tell you that this is not a clinical practice guideline, right? This is about engineering the device to be safe. And so we talk about things like what is the accuracy and how do you measure it? And what are the constraints when one measures the performance of the particular device. And from a regulatory perspective, it's my job to figure out whether your device works in a way that's, we use the term substantially equivalent, but that doesn't mean anything. It works in a way that acts just like the other devices that are out there. So if I pick up an oximeter and I use it, you know, and it tells me my accuracy is 2%, should be able to pick up another oximeter. And if its accuracy is 2%, I can use it, it's interchangeable from a clinical perspective. And our job in the standard is to try to identify what are these performance parameters that have to be controlled. And in fact, you know, when we take a look at the desaturation protocols that you've heard so much about, idealized laboratory studies, those are actually designed to eliminate all variability except changes in saturation, so that we can know that the devices are performing equivalently, at least at its heart, with the full recognition that when they get into the real world, they will behave differently. And I just want to talk a little bit about statistics. Did this go backwards? Yeah, so this is the formula for root mean square error. We don't have to talk about that. But we know that these devices are an estimate. And what that means is that for a given saturation value as measured by a co-oximeter, a pulse oximeter will have uncertainty. And so it could be plus or minus, one standard deviation, which might be plus or minus 2 or 3. It could be plus or minus 2 standard deviations, depends on how you want to look at this. But there's a lot of corrections made that people say, well, why don't you just slap up the confidence interval on the front dial of the oximeter? And then you'll know if it reads, 92, that it's 92 plus or minus 3. Well, the problem with that is it doesn't tell you what the accuracy is, right? What the underlying SaO2 value is, right? So we can talk about controlling variability, and this is the annex, the protocol, the clinical desaturation protocol, which is part of the standard and which the FDA guidance document recommends. And we do our best to talk about the inclusion and exclusion criteria. And you can see that skin color is one of the things that we talk about, and age and gender. And these are all under current discussions, and we don't yet have answers. There's lots of studies going on that we are hoping to empower valid conclusions about these. We talked about this, right? Ambient light, we know all these things screw up oximeters, and therefore, when we do the idealized laboratory studies, those are controlled, but they get published to say, hey, if you're a clinician and you're using these boxes, you darn well should be aware of the following issues, because your results may vary. And no discussion of oximetry would be complete if we didn't open up the table to what are the research possibilities. As Mike alluded to, there's lots of issues out there, not the least of which is, what is the statistic that we should be using to report the performance? We've been using the root mean square error because that's a combination of the bias and the precision that kind of gives you, the clinician, a single index, which we thought was digestible and which industry and clinicians have been using for, I'll say, decades. But maybe that needs to be fixed. Maybe there's additional information, and clearly we have a problem with reporting on pigmentation. That needs to be fixed. And just figuring out how to objectively measure pigmentation has turned out to be much more complex than just, slapping a scale on it or something. We can get into more futuristic things or more complex things, such as computational modeling to see where the photons are actually going. So there's lots of paths for research that can hopefully shed light on figuring out what the root causes of these errors are and ways that we can control them. And I thank you very much for your time and attention. Mike?
Thanks very much, Sandy and Bob. I'm just going to try and get my screen back up here. Give me just a moment. Actually, I'll just go ahead and let Carolyn Hendrickson start sharing your screen while I do introduction. So Carolyn, if you want to start pulling up your screen, we'll have you speak next, and then we'll do some discussion after Carolyn speaks to address Bob and Sandy's talk, as well as Carolyn's talk. So Dr. Carolyn Hendrickson is the site PI for the EQUIOX study as part of the Open Oximetry project, and she'll be talking about the EQUIOX study. Dr. Hendrickson is a pulmonary and critical care medicine specialist at UCSF, based at San Francisco General Hospital, where she's the medical director of the medical intensive care unit. During the pandemic, she's led a number of safety initiatives, clinical research studies, as well as clinical planning for much of our intensive care patients here in San Francisco who went through San Francisco General Hospital. Dr. Hendrickson today is going to be talking about harmonizing data collection for prospective real-world pulse oximetry studies. Thanks very much for joining us. Dr. Hendrickson, over to you.
Hi, Mike. Thanks for the introduction, and nice to be here with everybody. Are you guys seeing my presenter view screen or
Looks
my?
Great.
Okay, great. I'm doing dual monitors here. Okay, so like Mike said, I'm going to talk about our prospective study that we have started over the last few months at San Francisco General. And this presentation is meant to be sort of an overview of some of the issues we've been troubleshooting in case they are helpful to you. And this is also meant to be an opportunity for us to learn from you all who have been doing similar studies or related work to make sure that we're collecting data in the best, most usable way possible. So please feel free to interrupt. It's hard for me to see the chat while I'm presenting, so just come off mute and jump in, or Mike will let me know if you guys want to make some comments. So I have no relevant disclosures on this topic. So what we're trying to do in our study is standardize data collection of key variables and prioritize a pragmatic and parsimonious approach to these data collection that will allow us to merge data sets across studies. Now, different investigators may have a particular question they're trying to answer that requires more detailed data collection on a subset of key variables of interest. And this is meant to be something that can coexist with projects that are more focused on a particular question. We'd like to see all of these data represented in all data sets, regardless of the specific research questions, so that we can merge all of our collective efforts and understand some of the biases in pulse oximetry across skin pigmentation in a variety of patient populations. So you've heard about the Open Oximetry project, and the website is here. So you can find our protocols on this website. And so the slides are meant to highlight some of these aspects, but there's more details available on this website. So before starting a study like this, of course, all investigators need to have an IRB approved protocol. We used a modified informed consent protocol, which our IRB is comfortable with because we have a variety of prospective cohort studies with some biospecimen repositories from critically ill patients that are time sensitive. And so if we're collecting a small amount of blood, a minimal risk study has been approved in the past by our IRB, and they were comfortable with this proposal as well. So while we try our best to get informed consent before data collection begins, there are certain situations where patients come into the ICU, are maybe unidentified or don't have an identified surrogate and cannot consent for themselves. And we actually begin data collection while we're trying to identify the correct person to have a discussion with. We have relevant translations, and our IRB regulates this in a specific way. If we use a short form consent in a language other than English for more than 2 patients, we must pursue formal professional translation of those documents into that language. They take that as an indicator that our study population has enough of those speakers to need their own consent document that they are able to read in their preferred language. Of course, all equipment must be calibrated in accordance with manufacturer specifications. And we need to have safety protocols in place. Something that actually came up for us, I'm just calling it out here, is that we had an exclusion criteria eliminating research only subjects or samples in subjects with hemoglobins less than 8 grams per deciliter. This is because we have that waiver of consent. We need to have some safety mechanisms in place to make sure that we're not drawing excess blood on someone who's already quite anemic. And we had to revise our screening structures to make sure that this was highlighted, because we had a couple of protocol violations that we've worked through with our IRB. We needed to establish personnel training and reporting structures. And because patients can come into the hospital or the ICU at any hour of any day, we're trying to staff across multiple shifts during the day. And so we need to have a smooth handoff between the study personnel who may be collecting some samples under waiver. We want to make sure we're not over collecting on patients. And we want to make sure that we're holding up our agreement for informed consent, but not harassing patients and their families by, reaching out multiple times a day during an already stressful time. So we established a daily secure email for enrolled subjects that flag borderline or low hemoglobins for safety, that track sample collections. This is also available, of course, in our data collection website, but just as a kind of additional reminder to study staff, it's a consolidated view of that important information. We also highlight the end of the study window so that we don't exceed our 14-day window that the IRB has approved, as well as consent status attempts and follow-up plans. So we've chosen to use REDCap, and I'm sure that most of you are familiar with this really powerful tool. One of the reasons we chose it is that if you want to do a study similar to the one that we're working on, we can give you our data collection instruments, and you can use them in your own research and build on them to your particular needs. Similarly, if you have an awesome data collection tool and REDCap, we'd love to incorporate it into our work. Most of you know that it's HIPAA and 21 CFR compliant. And some of the packages actually have a remote consent function, which is really nice, especially during pandemic times when visitor restrictions are in place. So I found that tool to be really useful in some other studies. The basic materials and equipment that we have and that we're hoping will be standard across many studies is the pulse oximeters that are used as a standard of care in our ICU, or a Philips monitor. And we have a Masimo benchtop device for patients who have difficulty getting readings, normally because of low perfusion index, that the respiratory therapist will bring into the room in certain clinical scenarios. We also have a Masimo Rad-G handheld oximeter with a universal probe. We have a contact device, and these devices are meant to also represent devices that might be used in lower resource settings, and to be able to compare them to what's being used as standard of care in our hospital. We have a caliper to measure finger diameter, and we have a computer that's encrypted and password secured for HIPAA compliant data capture using REDCap. This is we have this laptop that rolls around on a mobile workstation and also holds all of our other devices. We have the Delphin skin color catch colorimeter. We have the Konica Minolta spectrophotometer. These were both mentioned in the chats about how to quantify skin color, skin pigment, and melanin and erythema indices. We are collecting Fitzpatrick data as well as von Luschen scale data and the Monk scale, which you heard about from Dr. Monk, and that there's been a lot of talk in the chat about this tool as well. Our goal was to capture skin color or skin pigmentation using a variety of modalities that would allow us to compare our data to what's previously been published, but also to try to improve the way we analyze this data and ideally identify actionable sources of bias. Here's just a visual of the data collection scales and tools that we use. Of note, the finger caliper, we actually think the analog version is better than the digital because there seems to be some more positional variability with the digital readout. These are our inclusion criteria. We study adults. They all have arterial lines in place for clinical purposes. And we say patients at risk for hypoxemia. So it doesn't mean you have to sat 90% to get enrolled, just that you have a clinical condition where we expect you might have some DSAT episodes. And we actually collect data on readings as high as 97% on the standard of care pulse oximeter probe. As I mentioned before, we do not collect research-only purposes for hemoglobins less than 8. We will collect clinical data that's ordered by the team for care purposes on these patients, but we won't draw any additional blood. We exclude people who are pregnant and people who are in custody of law enforcement. We have a routine screening list in our EMR. We have developed a tool in Epic that sorts by the lines, drains, and airways tab to identify patients with an arterial line. It also captures the most recent hemoglobin value as a flag to our coordinators about who to collect data on and when to pass for anemia. And then we also have a few subjects who might be identified by anesthesia providers in the operating room. The minimum data set required to contribute to our primary outcome is a paired SpO2 and SO2 sample. These, when I say paired, I mean we have a study staff a CRC or an investigator at bedside watching the ABG being drawn and recording the data from the monitors. We must have informed consent, or a permanent waiver of consent must be applied by a process that's been approved by our ARB, and that we've captured some of the time-sensitive data using this half-page printout, which would be happy to share with you. This just allows you to scribble down kind of what's on the monitor really quickly in real time at the time the ABG is being drawn. And includes things like vasopressor use, the mean arterial pressure, and in addition, relevant medications like vasopressors and neuromuscular blockers. So this is just a list of our most time-sensitive data that we're collecting. These are the things that we really encourage the CRCs to get right in that moment, and are focused on the SpO2 and the perfusion index from the standard of care monitor. If we have time to put the study devices, the RAD-G and the Contec on the fingers, we'll do that as well, but that's not our primary objective. We try to quantify the stability of the pulse oximeter tracing. Ideally, it's been reading about the same within a point or two for about 30 seconds. And then we try to describe the quality of the PPG tracing as well.
Sorry, do you only get PPG for the red signal, or do you get for red and infrared, or the other way around?
Sorry.
Are you only getting the
Sandy, you're breaking up.
You're only getting the PPG out of, I guess.
It's a single wavelength at present.
Sorry,
I'll type it into the chat.
hearing
Don't worry about
the
it.
question. Okay.
You're breaking badly, Sandy. Sorry.
Okay, we'll see the question in the chat momentarily, it sounds like. This is just a quick reminder that what we are collecting and what we hope other groups will collect is the functional saturation, the measured saturation on the arterial blood gas. This is not the calculated fractional saturation. So here are some challenges that we've run across. Stability of saturation, especially for these DSATs episodes, is rare because clinicians respond to it. And so they try to rescue the patient, which is the correct thing to do. So we know that these lower saturations are going to be not as well represented in our data points as some of the more stable and clinically targeted saturations. For that reason, we try to just be around, constantly walking around the ICU. We have a phone that the respiratory therapists and the nurses can call if someone's desatting and they're going to get a clinical sample. We can run over and try to be present for that data collection. We are noting the processing time from the draw to when the samples actually run on the oximeter. I'm sorry, the heme oximeter, and we are noting that as a key variable that we'd like to analyze to see if that is driving any biases in the data. And we are also interested to know if the manufacturer or the model of the blood co-oximeter impacts some of these assessments of bias. We use GEMS at our county hospital, and we're probably going to launch at UCSF Health, our tertiary care center, and they use the ABL machine. So we're curious if that might be a source of variance in our data. Oh, and sometimes the bedside devices can't read SAT, and we note that in our data set as -1 because the negative value helps flag that we couldn't get a read, and also if the absolute value is transposed in data analysis, 1 won't really register as anything meaningful, so it won't be mixed up. If, like, for example, if you did -99, you might get some data analysis errors. There are some challenges in having clinical research coordinators interpret ECG rhythms, for example, and so principal investigators need to be available to read these on the monitor or review printouts to make sure the data collection is accurate. And we also needed to establish a small difference between the atomic clock and the monitor's projected time in our hospital, just to if we're going back and doing data QI, we want to make sure, or quality control, we want to make sure that we've got the difference of 10 seconds built into our assumptions. We note patient movement, as I mentioned, vasopressor use, finger diameter, probe location, and then heart rate by both ECG and SpO2. So as I mentioned, all of our data collection protocols are here. I'm watching the time closely here because I want to leave time for discussion afterwards. So I'm going to move through these next few slides a little bit more quickly. As these are the tools, the scales and the devices that we're using. We are collecting the spectrophotometer and the colorimeter measurements at 9 different sites on the body. We're doing 4 sites for von Luschen, and then the perceived Fitzpatrick scale, sort of overall skin pigment. We're not asking subjects if they tan or burn. And so this is, imperfect application of that imperfect tool. And then for using the Monk scale, we plan to do the same 4 sites as we're checking for Van Luschen and also the cheek. So we are focusing on where the oximeter is doing its work. The fingernail, the distal interphalangeal joint between the nail bed and the DIP, the palmar surface exactly opposite of that. And we're also checking earlobe because these devices are often clipped onto the earlobe or sometimes the nair. And so we're checking skin pigment values at all of those sites. For the scales, we're using the inner upper arm as a non-sun exposed site that's commonly used in previously published data. We're also collecting data on skin tone of the forehead and cheek, as this is probably how a lot of cognitive processing and stereotyping and discrimination is applied at an individual level. To get these data, we close the blinds. We try to turn off as many lights as we can. Sometimes the nurses don't let you do that, but we do our best. We try to create a light seal between the skin and the device without pushing so hard that we're augmenting blood flow to that small site. We used a professional printing service to generate the resources the CRCs bring to the bedside so that the tones that are printed out are actually accurate to the intended scale. We laminate these cards and use a high-tech hole punch to make a deficit in the middle of each category so that we can have it adjacent to the skin and allow the reader to be as close as possible to the intended scale. All measures are taken in triplicate, and our plan is to use the median value. I mentioned earlier that finger diameter, we've found analog to be better. We're using the calipers to just lightly apply pressure without indenting flat. We found that there was a lot of inter-rater variability based on how much you squeeze the finger. So this is potentially a source of variance in data that we tried to address by getting folks to work together to agree on the correct protocol. And then I'm within 2 minutes of the end of my presentation, but I just wanted to say that we're collecting comorbidity and clinical data and created a REDCap instrument for this. We tried to focus on things that might impair perfusion and also impact tissue edema, as well as hemoglobinopathies or differences in skin pigmentation within the individual, like vitiligo. This is what it looks like in REDCap. We'd be happy to have any input after this presentation on if there's anything we missed or anything the other folks are collecting. We'd like to be inclusive with our data capture. So we plan to compare pulse oximeter errors between pigment categories. We're going to focus our primary analysis on the distal phalangeal sites. because that's where most oximeters are placed. And then we have some key mediators we'd like to include in our analyses to understand if they're driving any biases that we may detect. But we also plan for multiple secondary and exploratory analyses to look at skin pigment assessments from other sites and to understand what might be the most important drivers of the biases. We've enrolled 78 patients to date. We have 178 blood gas samples collected. We're targeting 250 to 300 subjects, most likely. This will be guided by an interim data analysis that's underway currently. And we're hoping to complete enrollment by 2023, by the summer of 2023. And then I'd like to just acknowledge all of the contributors to this work. It's a really great team to work with, and we look forward to including you as part of our team as well. So I'm going to stop talking and I'll stop sharing and see if we can get to the chat and also leave time for Mike's comments.
Thanks very much, Dr. Hendrickson. Appreciate that. I'm going to just pop up a few questions here on the screen to initiate. We'll do about 5 minutes of discussion, then we'll wrap up. Thanks everyone for bearing with us. There's a lot of chat conversation going on if you haven't been following that already. But in follow-up to Carolyn's presentation, one question is, we know of probably about 4 ongoing prospective real-world clinical studies that are either getting going or already underway, but we're trying to compile a list and potentially create a subgroup around that to communicate challenges and harmonize data streams. So if you know of ongoing studies, please drop them in the chat, either the PI or the site, and we can try and follow up. Or if anybody on this call has a study, please chime in now and share. We'd love to hear. And then a couple of questions just for the group based on what Carolyn presented. Are there data variables that you think we should be collecting? I see some folks have been commenting on in the chat. We'd love to harmonize that. You can also find our data collection protocols on the site, again, open for comment. But with that, we'll open it for commentary. Tarek, please go ahead.
Yes, thank you so much, Carolyn, for that. That's a really fascinating presentation. I had a question. Are you just collecting the end results, the pulse oximetry numbers from these muscles, or are you able to access the underlying data like the R values that they're collecting? I guess the companies would have had to give you access to them.
I'm sorry, I couldn't hear the whole question, but I think you were asking if we're just collecting what's projected on the monitor. Is that the question?
Yes, that's
yes.
right.
Yeah, so this is a pragmatic study. So yes, that's what we're collecting right now. We have some plans and a recent IRB modification to use a different data collection tool to collect our own PPG data with a research tool that's not part of the clinical monitoring system.
As a small follow-up to that, the group that I'm working with, we made a pulse oximeter. If you wanted to get all of the underlying data, we have units that you can plug into USB. I'm certainly happy to send you one if you want, so you get all of the R values, not just on the red and the infrared, but also on the yellow and orange, which gives you methemoglobin and carboxyhemoglobin. It might be interesting additional data in the future.
Oh, thank you for that very generous offer.
How many wavelengths? Sorry.
4. So red, infrared, and then I think it's yellow and orange. So basically carboxyhemoglobin and methemoglobin. Not quite the same as the Masimo ones, but similar.
Thanks for the offer, Tarek. And just to point out and follow up, on the website, we've posted the specifications for how we made our device from components that you can easily get access to, and a way to make it with multiple wavelengths as well. We would love to learn more about your device and kind of compare notes. That'd be great. Other comments? I see several folks chiming in with studies that are going to be starting. So thank you for throwing those in. We're going to keep track and definitely follow up with clinical studies, prospective clinical studies group to try and link folks. I think Carolyn already mentioned this, but we're very happy to share any documents to the extent they could be useful in accelerating your studies, protocols, data dictionaries, IRB, consent forms, And the other piece to add is another component of what we're working on, a little bit more relevant for some of anybody who's doing lab-based studies, is trying to create a diversity toolkit to try and improve diversity in clinical studies. That's being led by Dr. Bhumi Okanlola, who wasn't able to join today, but happy to share for those who are doing lab-based studies as well, as we try and address the issue of ensuring adequate diversity. Bob, I see you with a comment. Over to you.
Yeah, just a quick one, and I greatly appreciate the work that's being done in collecting real-world evidence. I would have to say that FDA has made the expectation in the past of manufacturers that they supply what is called convenience sampling. And Sandy can talk to this more specifically because the goalpost has moved a bit with that. And it doesn't always involve direct blood specimens, but at least something in the way of another reference device. But blood is often, if it's available, very helpful. And what has been found with most of those submissions, but more importantly, with some of the prior evidence of pulling together multiple different hospitals using multiple different co-oximetry devices, and this was specific just to NICU use on preterm infants, that the ARMS values can easily exceed plus or minus 10. So when a device is that imprecise in real world use, making decisions about what to do other than the child is certainly in a trench that you wish to extract them from, but fine tuning any titration of care, I want to emphasize these devices should be used with tremendous caution when trying to do something of that sort. Other direct measures are the gold standard. And this operating with a device that imprecise in a triage situation is troubling to me as a clinician.
Bob, thanks very much for the comments. We're coming up on time. I'm Dr. Edelman. I'll give you the final comments, and then I'll add just some quick summaries from my side and next steps. Over to you.
Just real brief, I just wanted to mention that we're in the process of updating our guidance. So all of the input that we're hearing today is extremely helpful, and we'll utilize it in updating our guidance. Thank you.
Thanks, Dr. Edelman. Thanks everybody for staying on for this meeting. Again, we hope this is really kind of the beginning. I think a lot of the comments have been very helpful in trying to formulate where we can go from here and how we can structure this forum. So we're going to take that to heart. We'll summarize and send out certainly the notes. We are asking folks to provide feedback on the protocols that have been referenced that can be accessible through the website, if possible, by January 6th of 2023. Sorry for the typo there. So, over the next month, we would like to welcome that feedback, and then we will try and create a new version of those protocols. And then again, as I had mentioned, what we are hoping to do is, as we're gathering input, we will send out a survey that will ask for input on priorities and ranking of priorities so that we can start generating some of those lists. We also will include a link to our new version of the data sharing platform and the consumer-oriented data sharing platform for device performance to welcome input to make sure that we're capturing the data that people are interested in and making it accessible in formats that are useful. We have not yet set the date for the February workshop, so stay tuned for some planning around that. It will be hybrid, so it'll be partly in San Francisco for those of you who are interested and able to be here, but it'll certainly be open to anyone to join, including those, and it'll likely be broken up into subgroups. Based on some of the discussion today and prior to today, there are several topics where there may be subgroups in the workshops and for asynchronous participation. So we're at the top of the hour. We're going to go ahead and have to wrap up. We'll
I raised my hand to ask if all the protocols that were presented are all being shared in that repository.
All protocols that are mentioned today are in that repository, yes.
Thank you.
Yeah, and if you're looking for something and unable to find it, please reach out to myself or Feker Nagusi, Fifi Nagusi. Both of our emails are in the agenda. Ms. Nagusi is coming on board as the program manager for the Open Oximetry project, and we welcome input and look forward to being in touch. So please Stay tuned for follow-up emails, and thank you again for your participation and time, especially to the speakers. And also thank you to the team here and the funders who have made the Open Oximetry project possible, including the Moore Foundation, the McGovern Foundation, the Robert Wood Johnson Foundation, the FDA, and USAID. Thank you very much. Have a great day.